Hello everyone, welcome to my channel Awesome Biology. Today I am going to discuss some of the previous year questions from the topic Biotechnology, Principle and Processes. So let's start. Question number 1. Which of the following is a restriction endonuclease? This question is coming in NEET 2016. Options were protease, DNase, RNase, HIN2. So protease is not a restriction endonuclease. Protease is a protein enzyme. DNase is a DNase enzyme. This is also not correct. RNase is a RNase enzyme. Not correct. This option also. Option number D, HIN2. Yeah, HIN2 is the correct option because HIN2 is the first known restriction endonuclease. So I will mark this with a green color. Highlight this. Okay, here option number D is the answer. Then question number 2. The cutting of DNA at a specific locations become possible with the discovery of. Options were given: restriction enzymes, probes, selectable markers, ligases. So the cutting of DNA, okay, is actually done by restriction enzymes, okay. So option A is the correct answer. So how the cutting is done, I will show you. So cutting is done on the DNA segment. Okay, this blue color segment is the DNA segment, and on that segment the restriction enzymes cut this way, have one cut and the other cut is on this one. So restriction enzymes cut the DNA fragment like this, and the segment between the two cuts were removed, okay, and on that gap desired segments were attached. So this process is known as restriction digestion. Okay, so the cutting of DNA at specific locations become possible with the discovery of restriction enzymes. So option A is the correct answer. Then option B is probes. No, probe cannot be correct because probes are the DNA segments that detect the presence or absence of DNA or gene. Okay, and C, to the selectable markers is also not correct. Selectable markers are actually the markers on the bacteria, okay, which are resistant to several antibiotics. And option D, ligases. Ligases are enzymes, okay, which are help in joining of the DNA fragment. Okay, so these all options are not at supportive to this question. So the correct answer is restriction enzyme. Then going to question number 3. Which one is true statement regarding DNA polymerase used in PCR? Options are given. Option A, it is used to ligate introduced DNA in recipient cell. So this DNA polymerase is not used to ligate introduced DNA in recipient cell. This option is false. Option B, it serves as a selectable marker. No, DNA polymerase doesn't serve as a selectable marker. Option C, it is isolated from virus. No, DNA polymerase is not isolated from virus. It is actually isolated from bacteria. Option number D, it remains active at high temperature. Yes, that is true. DNA polymerase remains active at high temperature because DNA polymerase is isolated from a bacteria that is known as Thermus aquaticus. So Thermus aquaticus is the bacteria from where the DNA polymerase is isolated, and that bacteria is temperature tolerant, that can tolerate a high temperature also. So that's why DNA polymerase used in PCR, it remains active at high temperature. So option D is the correct answer. Then going to question number 4. The correct order of the steps in polymerase chain reaction is option A, denaturation, extension, annealing. No. Annealing. Option B, annealing, extension, denaturation. That is also not right. Option C, extension, denaturation, annealing. No. Then option D, denaturations, annealing, and extension. Yeah, this is the correct order of steps in polymerase chain reactions. So I will show you how these steps takes place during the polymerase chain reaction. What happens? The DNA is actually used in the PCR is a double helical DNA. Okay, the double helical DNA is like this, and that DNA, when introduced in a PCR under the PCR, under the PCR, this helical DNA become heated, okay, heated in the PCR, and due to this heat, the DNA become denatured. Denature means disrupt or break, okay. The DNA breaks and separate from one another. This double helical DNA breaks, okay. So I will denote the first denaturation process as D, okay. So what happens after denaturations? The DNA double helical structure distort or breaks like this, and that distorted DNA separate, okay, into a single strand, okay. That double stranded DNA becomes single stranded DNA, and on that strand some primer attached to it. This pink color segments are the primers, okay, primers attached to it, and that condition is known as annealing, okay. Annealing, annealing, okay. I'm not writing the whole, so just I'm giving as a showing you as annealing. After that, after the annealing process, what happens? The extension of DNA takes place. So this, after annealing, the DNA lengthens, okay. Extension of DNA takes place. So this extension I denote it as E. Okay. So after the extension process, the last process is amplification. So in the amplification process, what happens? From the single strand of DNA, many copies of DNA were formed. So this is the amplification process. Okay, actually DNA polymerase is used for the amplification of the DNA segment, okay. Amplification means copy of DNA to produce from the single DNA to produce many more copies, okay. So actually the DNA polymerase is used for copying, okay. It's a Xerox copy, it's a Xerox machine. Simply we can say it's a Xerox machine for DNA, okay. It can produce many DNA segments from a single double helical structure of the DNA, okay. From a single DNA it produce many DNAs many DNA segments, okay. So from this we can easily understood the order. So first denaturation take place, then annealing take place, and then extension take place. So option D is the correct answer. So this question came in 2018, okay. This is a very basic question, very conceptual question, okay. And question number 5. The process of separation and purification of express protein before marketing is called. So the express protein before marketing, okay, some process, they undergo certain processes of separation and purification, and that process is known as, options were given: upstream processing, downstream processing, bioprocessing, post-production processing. So the right option is the downstream processing. So before marketing, the express protein undergoes separation and purification by the downstream processing. Option B is the correct answer. Then going to question number 6, which of the following is not a component of downstream processing? Separation is a component of downstream processing. Purification is also a process of downstream processing. Preservation is also a process of downstream processing. Expression, option number D, expression is not a component of downstream processing. So option number D is the answer here, okay. Right option. So question number 7, a foreign DNA and plasmid cut by same restriction endonuclease can be joined to form a recombinant plasmid using, options are given: EcoR1, Tech polymerase, polymerase 3, ligase. So as we know that the DNA and the plasmid or any DNA segments can be joined by the enzyme ligase, okay. So ligase joins two segments of a DNA. How it joins, I will show you. Assume this is the one segment of DNA and this is the other segment of DNA, and what happens? Ligase, what they do, ligase, this is the ligase, okay. This ligase joins the both segment of DNA, okay. This is it joins. This is the enzyme. The ligase is the enzyme. The joins the two separate segments of DNA. So the answer will be option number D. that is ligase. Question number 8. Stirred tank bioreactors have been designed for option purification of product, option B addition of preservatives to the product, option C availability of oxygen throughout the process, option D ensuring anaerobic condition in the culture vessel. So is a stirred tank bioreactors is a bioreactor where reactors were mixed. So where reactors or facilities of there is a facility, okay, for the reactors to mix with oxygen. So availability of oxygen throughout the process is required. So designed for the, it is designed, so stirred tank bioreactors is designed for availability of oxygen throughout the process. So option C is the correct answer here. Okay, option C is the correct answer. Then going to question number 9, the tech polymerase enzyme is obtained from. Options are given: Thiobacillus ferrooxidans, Bacillus subtilis, Pseudomonas putida, and option D, Thermus aquaticus. So tech polymerase is the enzyme which is obtained from the bacteria Thermus aquaticus. So Thermus aquaticus is the correct answer. Then question number 10, golden rice is genetically modified crop plant where the incorporated gene is meant for biosynthesis of. So golden rice is genetically modified crop plant, okay? It's genetically modified by recombinant DNA technology where the incorporated gene is meant for biosynthesis of, actually it is incorporated gene is meant for biosynthesis of beta carotene. Okay, beta carotene is a precursor of vitamin A. So here the right option is vitamin A, option number D. Then going to question number 11, commonly used vectors for human genome sequencing are tDNA, option A tDNA, option B back and yak, option C expression vectors, option D TA cloning vectors. So here the right answer is back and yak, that is bacterial artificial chromosome and yeast artificial chromosome are the commonly used vectors for human genome sequencing. Okay, then going to question number 12, which kind of therapy was given in 1990 to a 4-year-old girl with adenosine deaminase deficiency? Okay, options were gene therapy, chemotherapy, option B chemotherapy, option C immunotherapy, option D radiation therapy. So option A, gene therapy is the correct answer, because I will show you what is gene therapy, how it takes, how it occurs. Okay, the technique of gene therapy I will show you here. What happens in the gene therapy? The segment of DNA, we know that DNA is a double helical segment, okay, where in the segment adenosine, we know that in DNA there are nucleotides, okay, adenosine, at the nucleotides adenosine pairs with thymine and cytosine pairs with guanine. Okay, sometimes what happens, there's some misbase occurs due to some errors. Okay, misbase pair occur. This is a normal base pair pairing, adenosine pair with thymine, cytosine pair with guanosine is a normal base pairing. Sometimes mispair occurs due to some error, okay, in the genetic makeup. So this kind of mispair, such as adenosine have to pair with thymine, but instead of it pairs with guanine. So this is a misbase pair, okay, error. So this kind of error leads to some kind of diseases, which is found in a 4-year girl with adenosine deaminase deficiency. Okay, it's a genetic base pair deficiency. Okay, this kind of deficiency can be corrected by gene therapy. Okay, what in gene therapy do? Gene therapy remove this segment, remove this base pair, okay, or substitute this base pair, okay, and replace with a normal base pair, as a result of which the DNA segment and the base pairs were corrected. Okay, this correction was made by the process of gene therapy. Okay, so question was asked here, which kind of therapy was given in 1990 to 4-year-old girl with adenosine deficiency disorder? Okay, so gene therapy was given to that girl. Okay, okay. So this is the technique of gene therapy. Now going to question number 13. Here in the question number 13, the introduction of tDNA into plant genome. No, in the plant involves, okay. The introduction of tDNA into plant involves which of the following option? Infection of plant by Agrobacterium tumefaciens. Option B, altering the pH of soil. Heat shocking the plant. Option C, exposing the plant to cold for a brief period. Option D, allowing the plant roots to stand in water. So what happens? The tDNA, this is the tDNA is found in actually in pathogen Agrobacterium tumefaciens. Okay, this is a pathogen which plant pathogen. Okay, Agrobacterium tumefaciens is a plant pathogen, and it contains the tDNA. Okay, so when a plant is infected with a Agrobacterium tumefaciens pathogen, so the tDNA from the Agrobacterium tumefaciens enters into the plant. Okay, as a result of which the plants were infected with a disease, tumor kind of disease, and that disease is known as Crohn gall disease. Okay, so from this we can understand that option A is the correct answer, because the introduction of tDNA into plant involves the infection of plant by Agrobacterium tumefaciens. Okay, so option A is the correct answer here. Then going to question number 4, the process of RNA interference has been used in development of plant resistance to. So the process of RNA interference technology has been used in development of plant resistance to nematodes. Okay, option A is the correct answer here. And going to question number 15, continuous addition of sugar in feed batch fermentation is done to produce methane, obtain antibiotics. Option C, purify enzymes. Option D, degrade sewage. So continuous addition of sugar in feed batch fermentation is done due to purification of enzymes. So option C is the correct, to purify enzymes. Okay. Then coming to question number 16, the two polypeptides of human insulin are linked by option A, Phosphodiester bond. Option B, Covalent bond. Option C, Disulphide bridges, and option D, hydrogen bonds. So the two polypeptides of human insulin are linked by disulphide bonds. Option C is the correct answer. Question number 17. The first human hormone produced by recombinant DNA technology is insulin, estrogen, thyroxine, progesterone. So the first human hormone produced by recombinant DNA technology is insulin. Okay, something we have to remember. Okay, some questions we have to remember. Option A is the correct. The DNA molecule to which the gene of interest is integrated for cloning is called transformer vector template carrier. So DNA molecule to which the gene of interest is integrated for cloning is called vector. Okay, so I will show you how the vector is to look at. So vector is a circular DNA. Okay, so DNA molecule to which gene of interest is integrated. So gene of interest is the red color segment which is integrated to the circular DNA. Okay, so, and that makes a vector. So that is the option will be, the correct answer is the vector. Now going to question number 19. PCR and restriction fragment length polymorphism are methods for option A, study of enzymes. Option B, genetic transformation. Option C, DNA sequencing. Option D, genetic fingerprinting. So PCR and RFLP, that is restriction fragment length polymorphism, are methods for genetic fingerprinting. So option D is the correct answer here. Now the last question. Which vector can clone only a small fragment of DNA? Options are given. A, bacterial artificial chromosome. Option B, yeast artificial chromosome. Option C, plasmid. And option D, cosmid. So for solving this question, we have to go for the sizes of these all vectors. So we have to know the sizes of all these vectors. Okay. So the size of the first option, the bacterial artificial chromosome is 300 to 350 kilo base pair. Okay, AB, the kilo base pair. Okay, then yeast artificial chromosome. The yeast artificial chromosome has a size of, so this is a vector has a size of 1000 to 2500 kilo base pair. Okay, then option C, plasmid. Plasmid has a size of about 10 kilo base pair. Okay, then at the last, cosmid. Cosmid is a vector that has a size of about 45 kilo base pair. So from all these sizes, the smallest size is the plasmid. Okay, the smallest vector is plasmid with a 10 kb p. Okay, with only 10 kilo base pair. So the smallest vector can clone only a small fragment of DNA. So option C is the correct answer here. So option C is the correct answer here. Option C. Okay, this option C is the correct answer. Last minute. So hope you are benefited from these MCQs that came in previous years, and this pattern of MCQs have a maximum probability of coming in the upcoming NEET exam, because questions are repeated, okay, and patterns of the questions are repeated time after time. Okay, so be prepared, and please subscribe to my channel to get more and more videos like this. Thank you very much.